Hello everyone, I'm speaking on behalf of the Sainsbury Laboratory team that is attempting to come up with a blight-resistant potato. So we work at the Sainsbury Lab, which is originally still funded by David Sainsbury's private charity, and we also win grants to work on how plants and microbes interact with each other, how plants are resistant to disease and how microbes circumvent those resistance mechanisms. So one of the diseases we work on is potato late blight, which is a devastating potato disease. It causes worldwide $7 to $10 billion a year of losses or requirements for fungicide applications. And in a very short period of time, you can go from a field of healthy potatoes to a field of potatoes that have been wiped out if you don't control with fungicides. And typically UK or Northern European farmers spray 10 to 15 times a year more in a very wet year. And this disease was the cause of the Irish potato famine, or at least the biotic cause. And there, if you've been to Dublin, you'll see this rather moving monument to all the people who were casualties of that famine. It reduced the population of Ireland from 8 million to 5 million in the 1840s, both from deaths and from emigrations to the US. And it requires 50 to 6 million pounds a year to be spent on crop protection chemicals. And basically, we think we've got a solution, which is disease resistance genes. And what we did a lot of genetics. This is what we have a lot of experience at. And we identified wild relatives of potato that carry genes for resistance to this disease. And one of them, we've got one wild potato shown here. I'll just the laser pointer symbol. So we've got a wild potato, one potato wild here, wild potato here called Solanum venturii, and there's another wild potato here, which looks a lot like Solanum nigrum, black nightshade. That's because it's its ancestor. It's called Solanum americanum, and we got resistance genes from each of them. So one from venturii and two from americanum. And when we put those in to cultivated potato Maris piper, and just plant the transgenic line that's received these genes next door to lines that haven't received these genes, you can see that the Maris piper that doesn't receive these resistance genes is wiped out by blight, whereas the one that's received these resistance genes is not wiped out by blight. Now we have a collaboration with a US company called Simplot, which has developed a commercial trait that improves tuber quality. And we've also got a commercial partnership with this embryonic British startup called BioPotatoes. In the course of this, we've also added to this resistance set of cassettes genes that silence two genes that give you poor tuber quality. So if you silence this one called PPO, polyphenol oxidase, then you don't show bruise damage. And if you silence this one here, invertase, then you don't convert sucrose to the reducing sugars, glucose and fructose. And that has benefits which I'll be telling you about on the next slide. This has been funded by the British taxpayer. Thank you very much, over the last 20 years. And we finally got a product out of it. So yeah, this illustrates the difference between the susceptible and the resistant transgenic lines. I should say that what we've done by putting in genes is something we cannot do by gene editing, about which you might have heard a lot recently. We're using another entirely benign method called the GM method, by which you can put in genes that confer a useful property on the crop. And what genes we're putting in, I should add, they confer resistance because they help the plant turn on its own plant defenses in time to stop the pathogen succeeding. So these kinds of molecules are called receptors. They basically like their antennae for the plant. The plant after our addition of genes, has an enhanced capacity to perceive when a pathogen shows up, or when late blight pathogen shows up, and activate defense. Now, we've tested these lines for a number of traits. We've tested them to show that their yield appears comparable on a very small scale trial. We've tested them for glycoalkaloids. There's no elevation in glycoalkaloids that some potato varieties can have through bad luck in the breeding process. An additional trait is that there are a lot of losses in potato to late blight in storage as well as in the field. So if you've got regular Maris piper here, wild-type Maris piper, and you infect the tubers with late blight, 2 weeks later, this is what they look like, and you can't sell them. In fact, you can't eat them. This is an uninfected control. These lines to which we've added these receptors so they can perceive when the pathogen shows up and activate defense are fine. They just shrug it off. So they've got tuber blight resistance as well. And in addition, because of those traits in collaboration with Simplot, we've got reduced propensity to bruising. And we verified that for our lines, that those genes are working. So you can lose a lot of potatoes in production. This gives you food wastage, spoilage, and with this trait, you don't have those losses. And also, if you've got these more reducing sugars, which particularly accumulate when you store potatoes at low temperatures, like 3 degrees, if you do this with regular potatoes and you cook them at high temperature, this is what they look like. Who's going to want to eat these? But if you store our potatoes, our improved potatoes, for 6 months at 3 degrees, then this is the sort of property you see when you make crisps out of them. So yes, this bruising causes post-harvest losses. Cold-induced sweetening also gives you losses. And one of the advantages of this trait here, the reduced accumulation of reducing sugars on cold storage, is that it means that you can store the potatoes colder to stop them sprouting, and that means you don't need to add this rather nasty compound CIPC, chlorpropan, which has actually just recently been removed from being permitted to reduce sprouting in stored potatoes. And of course, we think this is a good thing to do, and love him or not, our Prime Minister has made positive remarks about the kind of work we're doing. When he came to the steps at 10 Downing Street to enter as Prime Minister for the first time, he said, "Let's start now to liberate the UK's extraordinary bioscience sector from anti-genetic modification rules. Let's develop the blight-resistant crops that will feed the world." And I think he was thinking of our blight-resistant potato when he said that. So we'd love to see the UK be a leader in innovative biotechnology for agriculture, both editing and GM. There's a lot of beneficial traits in addition to the ones that we've been working on to evaluate and deploy if they work. And we'd like to see which ones are actually useful in the UK, in addition to those that we've told you about just now. Now, we don't think GM's a magic bullet for solving everything. Back in the days when people worried about, started to worry about energy security and climate change, you saw people arguing about whether we should solve the energy problem by nuclear or better insulation or more efficiency or more renewables. And actually, this is a silly argument. You need all of them. And in the same way for food security, you could argue about whether we should achieve it by reducing meat consumption, reducing food waste, better agronomy, or different kinds of better genetics. You need all of them. But of course, we need breeding, and we need to use the GM method, and we need to use editing. So we need every tool in the toolbox for sustainable food security. So that's told you a bit about our work and our achievements. Here's the team who got us to this position in the first place, and Agnieszka and Sebastian are carrying it on. Gerard Bishop is a collaborator at NIAB, and you can see a video at this link where he tells you about the field trials they're doing on our potatoes at NIAB. and you could send email questions to us at blight-trial@tsl.ac.uk, and thanks for your attention.